This is podcast 15. So we, in the last podcast, talked about glycolysis. At the end of glycolysis, uh, you're going to either continue oxidizing pyruvate in mitochondria, or you will go to fermentation. So that all depends on the availability of oxygen. So in most cases, I'll talk about a special case where it doesn't matter. In most cases, uh, in all cases, glycolysis can happen with or without oxygen. It does not directly depend on oxygen, so it's a source of energy for cells whether or not there is oxygen. If there is oxygen around, you can get a lot more ATP out of that pyruvate molecule, and we'll talk about that in future podcasts. If there is no oxygen around, there still is opportunity to get ATP out of glycolysis. Organisms that require oxygen to meet their energy demands are called aerobes. Organisms that can live without the presence of oxygen and meet their energy demands are called anaerobes. If you have to use oxygen in order to meet your energy demands, you're considered an obligate aerobe. If you can or cannot, depending on the conditions, that's called a facultative. So if there's oxygen around, I'll be an aerobe, but if there's no oxygen around, I'll be an anaerobe. That would be considered a facultative aerobe. Humans are considered obligate aerobes, even though there are cells and conditions within humans that can operate anaerobically for a short amount of time. Okay, and this is the pathway that we'll pursue in another podcast, the next podcast, when we're talking about further oxidation of pyruvate. Okay, so there's no oxygen around, and you can't further oxidize pyruvate by aerobic respiration because there's no oxygen around. So what do? Depending on the type of organism you are, you can do fermentation, which allows you to convert the pyruvate into lactate or the pyruvate into ethanol, depending on the organism that you are. And what this does is it allows NADH to be regenerated. So you form, NAD to be regenerated. So you form NADH during glycolysis, and NADH is then eventually going to be reoxidized during electron transport. But electron transport requires oxygen. So there's no oxygen around. You want to keep on doing glycolysis so you can get your measly 2 ATPs per glucose energy, and there's no oxygen around, so that's better than nothing. But there's a limited supply of NAD to do that. So you have to, in order to keep glycolysis going, you've got to regenerate NAD. So this pathway takes pyruvate. It actually reduces pyruvate with the hydrogens from NADH, regenerates NAD, and makes lactate. The enzyme that does this is called lactate dehydrogenase. In the pathway to ethanol, you can have pyruvate decarboxylase plus alcohol dehydrogenase, eventually making ethanol. And again, you regenerate the NAD, which will be used in glycolysis. So before this slide, you had all the pathways in glycolysis leading to pyruvate. If you want to keep that going, you've got to take away the end product, pyruvate, and you've got to regenerate NAD. in order for this to keep glycolysis going. That is, if there's no oxygen around. So depending on the organism, you can make lactate or you can make ethanol. Yeast can make ethanol. This is important in fermenting bread, causing bread to rise. The CO2 that is given off in this process causes makes bubbles in the bread, causes the bread to rise, and then the ethanol is burnt off in the baking of the bread. When you make beer or wine, the ethanol stays in solution, and that's part of the desired product of the yeast. In making yogurt, you make lactic acid or lactate, depending on if it's got a hydrogen on it or not, and that's a desirable product of the microorganisms making the cultures making the yogurt. And you also make lactate in the situation where you have run out of oxygen, such as you overused your muscles past the point in which you can deliver oxygen. You're using oxygen faster you can deliver it. You're left with glycolysis, and you build up lactic acid in your muscles. Okay. So NAD is in short supply in cells. If you didn't regenerate the NAD, you would not be able to continue to do glycolysis. So these pathways, taking pyruvate to either lactate or ethanol, allows you to regenerate that NAD. And these are just the two most common pathways that we run into in organisms that are typically studied. But microorganisms can do all kinds of other pathways leading from glycolysis to regenerate NAD. Okay. Here in this diagram here, you've got the sort of backbone of glycolysis, or at least parts of glycolysis. And the idea here is to show you that lots of other things can enter glycolysis. So here's glucose going to glucose 6-phosphate, and then fructose 6-phosphate, and then fructose 1,6-bisphosphate. That's glycolysis you know. But you could also start with fructose and go to fructose 6-phosphate. You can go start with sucrose, which is a disaccharide, and then split that into glucose and fructose. You can start with lactose, milk sugar, and split that into galactose and glucose, and then convert galactose into glucose 1-phosphate. Later on, we'll talk about breakdown of fatty acids. The most common type of lipid that you store as a human is called triacylglycerol. It has a glycerol backbone, and when you take off those acyl chain groups to break down in beta oxidation, again, we'll talk about that in the future, you can take the glycerol and then have it enter glycolysis at this pathway. Okay? So the point of this slide is not to memorize everything on the slide, but to recognize that glycolysis is a hub for other intermediates entering this pathway. You don't always have to start with glucose to go through part of glycolysis. And you can also regenerate glucose from other intermediates, typically lactate or depending on the organism, ethanol. But most of the time we're talking about, in humans, we're talking about lactate. So you have a bout of exercise, you build up lactic acid because you had low oxygen tension in your muscles, and now you are resupplying your muscles with oxygen, and you want to get the energy that is still in that lactic acid out of that lactic acid, and so you regenerate glucose to go through glycolysis. So in regenerating glucose, you're going to have to add energy to it. Here's glycolysis on this side. Here's gluconeogenesis, glucose. new genesis, making new glucose. That's what this means. Okay. Many of the steps, 7 out of the 10 steps here, are the same enzymes that you would see in glycolysis. They're just in reverse. However, there are 3 steps catalyzed by 4 different enzymes, because this one is broken into 2 enzyme steps, where in the glycolysis direction, the reaction is so spontaneous, has such a high delta G, that going in the reverse direction is thermodynamically really hard. So it's really spontaneous in this direction, going toward the bottom of the page. So going in the other direction, going toward the top of the page, difficult thing to do. And so there are these sidestep reactions when we're going from pyruvate back to glucose. And they are catalyzed by enzymes that are using ATP or using GTP, which is just like ATP, it just has a different base, and releasing the potential energy in that terminal phosphate and adding that energy to the reaction to make the reaction overall spontaneous, going in this direction. You can see here, here, and here where energy is added, and here where NADH is oxidized back into NAD, whereas during glycolysis it's the opposite. NAD is reduced into NADH. So the idea is that if you're in a situation where there's not oxygen around and you have to regenerate NAD, you may do fermentation and generate lactate, for example. When oxygen comes around again, you don't want to waste all that energy that is still left in lactate. You can still get lots more from it. And so you convert that lactate back into pyruvate, and then that pyruvate back into glucose to do gluconeogenesis or regenerate glucose, and then you can get lots of that energy back out of that glucose molecule. Much of the gluconeogenesis pathway, going from the bottom of the page to the top, is very similar to the glycolysis pathway. 7 out of 10 enzymes are the same, but for 3 of the steps, it's very spontaneous, very high negative delta G going in the glycolysis direction. So going in the opposite direction, you have to add energy in order to make the overall reaction spontaneous, going toward gluconeogenesis. Okay, and as I said before, a situation where this might happen may be that you generated lactate in your skeletal muscle because you were sprinting and you used up oxygen in those muscle cells faster than you could supply oxygen. And so in order to keep glycolysis going and regenerate NAD, you made lactate. Then after that bout of exercise, you pump that lactate out into the blood, and it's taken up by the liver, where there's gluconeogenesis happening and glucose is being resynthesized starting with lactate. So they can be in different compartments in a body. There are many ways to regulate glycolysis. As I mentioned when we started talking about glycolysis, that it's still very heavily studied. It's still an important mechanism in cancer and also gluconeogenesis. And there are known points, are known intermediates that control the flow of the flux of intermediates through the pathway. So if you want to know how fast glucose is being oxidized to pyruvate in glycolysis, or how fast pyruvate is being reduced back into glucose in gluconeogenesis, there are known intermediates that influence the rate of that pathway. So the concentration of glucose 6-phosphate is a negative effector on hexokinase, which is which catalyzes first reaction to make glucose 6-phosphate. So the more of this you make, it is allosterically inhibiting hexokinase. So the more you make, and it'll shut down this enzyme. PFK1 is negatively regulated by ATP and by citrate. We're going to see where citrate comes in the next podcast, but is positively regulated by AMP and F2,6BP, bisphosphate. So these are things that would show up if ATP was low. These are things that show up when ATP is high. If there's ATP is high, then you don't need to do more glycolysis. If ATP is low, then you do need to do more glycolysis. That's the idea behind this slide, is that there are many allosteric effectors of this pathway. So in addition to the rate limiting step things, you can measure the concentration of these intermediates to get an idea of, is glycolysis being inhibited, or is glycolysis being enhanced? Here's an H5P question for you.